Hi everyone, I'm Hannah Cachadorian. I am a rising junior at Worcester Polytechnic Institute and I am a biology major on the pre-med track. And today we're going to talk about Rosalind Franklin and her impact on biology and genetics. Rosalind Franklin's work changed the world of biology in all of its underlying specialties. Her efforts to better understand the DNA molecule provided invaluable insight into the way genetic information is stored and replicated in all living things. This is especially important because DNA is known as the central dogma of biology. Essentially, DNA is the main idea that all biology revolves around, and by having a strong understanding of DNA, we are able to understand all of the other aspects of biology. Without Franklin's work, we wouldn't be able to do all the amazing work that we do each day, whether that's in medicine, ecology, zoology, microbiology, molecular biology, or any other biology concentration you can think of. Rosalind Franklin was a British physical chemist that specifically discovered the structure of the DNA molecule. By using a special X-ray, she was able to obtain a picture of what DNA looks like, allowing her to make the conclusion that DNA molecules were a double helix shape. Using this information, she was also able to determine that according to the way the nucleotides, or the rungs of the ladder if you can imagine the shape of DNA as a ladder, were connected, the two strands of the double helix were complementary, which allowed for DNA to be replicated. In addition to her work with DNA, she was also able to lend some insight on viral genetic information and virus structure. Her work here also aided in laying the foundation for the field of structural biology. Before working with DNA, Rosalind Franklin studied X-ray diffraction in Paris under a French scientist. Her work with X-ray diffraction was later applied to her work with DNA when she was researching DNA at King's College in England. Using the X-ray methodology, she was able to obtain an image of the DNA molecule. The data acted as a springboard for further studies that confirmed the structure and function of DNA as we know it today. To obtain the image of DNA, an X-ray was projected onto a DNA sample. The diffracted rays of light create a diffraction pattern on a detector placed behind the sample. The diffraction pattern that was projected onto the detector is now known as photo 51. This is photo 51. It doesn't look like much, but what seems like a funny dashed X actually provided enough information to Franklin that she was able to piece together what a DNA molecule looks like and how it has the ability to replicate. This is an image of a DNA double helix. This is the model of DNA that we use today, and it's the image that pops into your head when people talk about DNA. Rosalind Franklin was responsible for bringing this model to life and providing scientists with the building blocks to further understand life and genetic information. Franklin is most famous for her work on DNA, however, she has also provided insight on viruses. Through her work, she was able to determine where viral genetic information was located and how it exists in the virus. Franklin discovered that the genetic information is in the form of RNA, which is like DNA but with only one strand, so it's kind of like a ladder that's missing the second half. She's also discovered that RNA is not present in the viral central cavity, but in the protein of the virus. I wanted to close this video out with a quote from Rosalind Franklin herself. "Science and everyday life cannot and should not be separated." I think this is an extremely important sentiment, as science helps us understand everything in the world and everything in our day-to-day lives. Without science, we wouldn't understand much about the world around us. Science is much more than a school subject. It's a discipline filled with curiosity and discovery, and I hope Rosalind Franklin's story was able to inspire you just a little bit. Thank you for watching.